This is going to be the last video in the series preparing for Exam 2, and just so you're following along in the notes, the information on controlling microorganisms through things like temperature and UV light and desiccation, that stuff's so straightforward. I'm just going to leave that for you to read over in the notes. And so I'm going to jump to starting off on basic principles of antimicrobials. And terminology-wise, this involves drugs that are designed to target bacteria, eukaryotes like fungi, antifungals, antiviral medications. The principle's all the same. We're going to be mostly relating this to bacteria in this section, and we'll move on to the others in future exams. But again, the basic idea is all the same. You want to find a drug that inhibits something or blocks something very, very specific to that pathogen, to the bacterium or the virus or the yeast, whatever it happens to be. And the purpose of that is you can't give a person a drug that's general, because the things that are generally common between all different organisms, bacteria, fungi, even humans, we all have cell membranes. All organisms operate based on transcription, translation, and using proteins for their primary functions. And so you can't give a human a drug that just disrupts cell membranes or damages DNA or disrupts translation. You can't do things like that because it'll just kill the person. And so the specificity part of this means you want to find a drug that blocks something specific. It's most commonly an enzyme, but not always. Could be a cell receptor on the surface of the pathogen. And ideally, you want that drug to only be found in the pathogen that you're targeting to make it as specific as possible. That way your drug is precise and you're not having off-target effects. So you can use, for example, antibiotics that bind ribosomes. We can get away with this because, in some cases, because bacterial ribosomes are substantially different from human ribosomes. They're doing the same thing, translating RNA into protein, but their structure is slightly different. You could use an enzyme that's specific to the pathogen. If you block the enzyme from binding its substrate, it's not going to be able to carry out the reaction. In viruses, often we use medications that target an envelope protein on the surface of the virus that'll prevent it from attaching to whatever cell inside the host it's trying to enter. And in the end, it doesn't really matter which target you go after. The important thing is just to find one that's effective, that you have a drug that will work well on. Okay. A little more information before we keep going. An interesting question is, when you go to a physician's office because you have an infectious disease, antibiotics are common. They're prescribed regularly, really prescribed too much. But the times when they are effective and are needed, they usually, not always, but usually do their job fairly well. And it's an interesting question. Antibiotics and antifungals, killing yeast and fungi is pretty easy. Why are those fairly easy to develop? But antiviral medications, they do exist, but not as commonly, and they're usually not as effective. And a couple reasons for that is, if you think about it, the goal of an antimicrobial drug is just really to kill something. You're trying to break a biological machine. Bacteria and fungi are simple compared to humans, but they're incredibly complicated compared to a virus. And so part of it is bacteria and fungi just have more parts. Right? They have more enzymes, they have more receptors, they have more targets that you can go after. And so you have more options to work with. The other thing is viruses don't have a metabolism. It makes it very hard to kill something that's not truly alive. And so antibiotic medications can actually be bactericidal or fungicidal. They'll actually kill the cell that you're targeting. With a virus, you can't really kill it without denaturing protein. And since you don't want to denature human proteins, that's out of the question. And so all you can really do with a virus is slow it down. You can't actually kill the thing off. Okay. Looking at it from another direction, you could say the goal of all of this is to find a target that's pathogen specific that you don't find in a human. Okay. Bacteria, viruses, fungi, they all fall into this category. They're different enough from humans. It's pretty easy to make drugs to go after. Viruses are also very different, but like I said, they're just too simple. On the opposite spectrum, I think it's very useful to compare antimicrobials to chemotherapy for cancer. Cancer is very complex, right? It's easy to find drugs that will kill cancer cells. The problem is they're so incredibly similar to healthy human tissue that it's difficult to find a drug that will kill the cancer but won't kill the rest of the patient's cells. And so you've got these two variables that you're weighing. It's easier to kill complicated things, but you also need them to be very, very different from humans. Bacteria occupy that sweet spot where they're complicated enough to be easy to kill, but they're different enough from humans that you don't have to worry about crossover effect as much. Okay, so on that same idea, we can put a number on that, hypothetically at least, and call it therapeutic index. And what the therapeutic index really is a ratio of toxicity to effectiveness. And when, in the examples, when we say toxicity, we mean to the patient, human toxicity. And effectiveness, we mean at targeting the pathogen, bacteria in our case. Okay. All this means is, and it seems like common sense when you look at it on its face, you want a drug that has the highest toxic threshold to humans, meaning you need a really, really large dose to be toxic, and you want a drug that has the smallest effective dose for killing bacteria. So in this example, we're saying it takes 50 micrograms of a drug to be toxic, to be dangerous. It only takes 5 micrograms to be effective. Right? 50 divided by 5 equals 10. Okay. This just means, rephrased the other way, you would have to overdose to 10 times the effective dose to become toxic. Okay. In this example, toxic dose is only 40, so the toxic dose is lower, but the effective dose is only 1 microgram. 40 divided by 1 is 40, and so you would have to have a 40x overdose to start becoming toxic. And so the bigger the therapeutic index, the better, because it means it's giving you more cushion. You have more room to play with where the dose could be ramped up a little higher to be more effective at killing the pathogen and not have to worry about the consequences of toxicity in a person. The other side of this, though, is it's not quite that straightforward and simple. The terms effective and toxic aren't straightforward. Does effective mean slow down the infection a little bit? Does it mean kill off 90% of the bacteria in an infection site? Does it mean kill all of them? What does toxic mean? Does toxic mean gives you an uncomfortable rash for a few days, or does it mean causes a heart attack, or does it mean causes blindness? What does toxic actually mean? And so in practice, this is a lot harder to calculate because you have to decide exactly what are you calling effective and exactly what are you calling a toxic or harmful dose. Okay, so moving on, I want to talk about a handful of specific antibiotics, and there are hundreds of variations out there and available for use. We're only going to go for a few because obviously we're constrained by time. So a little bit of the background, and this is important for understanding sort of how and why drugs are developed and what the strategy is. Antibiotics are almost always derived, at least in the beginning, from natural sources. What it means is they're metabolic byproducts of either bacteria or fungi. And this makes sense because bacteria and fungi are in competition with each other. And when you have multiple organisms growing in close proximity, they're going to try to, I shouldn't say try, natural selection is going to favor the production of antibiotics that kill off their competition. Okay? And the best example of this, the ones that almost everyone has heard of, are molds that produce what we call penicillin. And the purpose of the mold producing penicillin is it gave them a selective advantage by killing off bacteria that are competing for the same nutrients that the mold is. Okay. And the reason this is important is what it shows you, developing a brand new antibiotic is incredibly difficult. Okay? So instead of having a chemist or a chemical engineer take the time to try to make a brand new antibiotic from scratch, it's been much, much more successful to go out into the natural world, look at bacteria and fungi that produce their own antibiotics that they've developed through millions and millions of years of natural selection. Right? It's given us a lot of good drugs to work with. Okay. From there, chemists can do their work by modifying. And so when you see penicillin and ampicillin and methicillin, all those antibiotics that end in "-cillin, those are derivatives of penicillin. They've been tweaked, they've been modified. We can play with it a little bit. But coming up with the original baseline drug that we're jumping off of, that's almost always best left to nature. All right, so penicillin is one example, and we're going to talk about that whole category of drugs in a little bit. This is an overview of the most common targets of antibiotics. And the two that we're going to talk about are cell wall inhibitors and protein synthesis inhibitors because they're the most common. There are drugs that target cell membrane integrity, but again, those are hard because humans have cell membranes, so you have to find something unique to bacteria. You can find drugs that target DNA and RNA, but that's really hard too. They often don't work incredibly well because the polymerases, the enzymes that copy DNA and RNA, are fairly similar between bacteria and humans, so those don't work incredibly well either. And folic acid synthesis, it's just a small pathway, so there aren't a lot of targets available. And so we'll mention cell wall inhibitors, beta-lactams specifically, which is the penicillin family. And we'll mention some protein synthesis inhibitors too. Right. Every drug has a certain spectrum of effect. And all that really means is some bacteria or some antibiotics target a very narrow spectrum of bacteria, maybe specifically, gram-negative bacilli, whereas others target a very broad spectrum. They can kill almost anything in the right dose. And it's not an all or nothing thing. You can't simply say this antibiotic is broad, this one's narrow. It's a whole sliding scale, a range of effect that they can have. And I want you to think about what the benefit of each is. It really comes down to two things. One is, do you know exactly what you're trying to kill? Which means, has the pathogen been identified? If you know what the pathogen is, and you have an effective narrow-spectrum antibiotic available, it's always the best bet. Okay, so if you know what you're after, and you have a drug that'll do the job that's narrow, that's a great thing, because then you're not going to go and kill other bacteria, normal resident microbes, especially in the GI tract, that could be harmful to the patient. That doesn't happen very often. And so broad-spectrum antibiotics are necessary in one of two cases. The most common is, if you don't know exactly what the pathogen is, you can't use a narrow-spectrum antibiotic because you don't know which one to choose. The other is, sometimes a narrow-spectrum option isn't available. Right? There are only so many antibiotics out there, and some narrow-spectrum options that we would like to have just haven't been developed at this point. Okay. So narrow spectrum, always better if the situation is aligned to it. Broad spectrums are more often used because both of these conditions aren't met. You either don't know what it is or you don't have a good option available. Okay, so the first category of drugs we're going to look at are those that target cell wall synthesis, and all they're really doing is interfering with an enzyme that's required to synthesize peptidoglycan. Whenever a bacterial cell divides, it has to make a whole new batch of peptidoglycan to form the cell wall of the new daughter cell that's being produced. If not, then it's susceptible to osmotic stress, the membrane's left unprotected, and the cell will just lyse. And these drugs called beta-lactam antibiotics, that's the most common ones are in the penicillin family, they react with one of the enzymes in that synthesis pathway to turn off, or at least dramatically turn down, the production rate of peptidoglycan. And the way that works is Beta-lactam antibiotics, each antibiotic is slightly different. They're different molecules, but what they share in common is this structure highlighted in red called a beta-lactam ring. That's the actual substructure within the molecule that binds to that peptidoglycan synthesizing enzyme and turns off its ability to produce cell wall. The consequence of that is they're pretty broad spectrum. All bacteria have to produce peptidoglycan, and so they'll have a pretty strong effect no matter which bacterium you're fighting, with a few small exceptions. So the only downside to this is these beta-lactam antibiotics, like I said, have been existing in organisms like molds for a really, really long time, millions and millions of years. That's given a long time for natural selection to run its course. The consequence of that is that some bacteria have evolved an enzyme called beta-lactamase, that's this guy, that can destroy the beta-lactam ring and inactivate the antibiotic. Okay? It's a defense mechanism. If the bacteria can produce an enzyme that destroys the antibiotic won't have an effect. And so when we talk about bacteria being resistant to an antibiotic, penicillin resistant for an example, all that means in this case is they have a gene somewhere, either in their chromosome or on a plasmid, that codes for beta-lactamase. And when you give the antibiotic goes into the patient, it goes inside, diffuses inside the bacterial cell, but if this enzyme just destroys the antibiotic before it can have its effect, they can still make their cell wall and go about dividing just like they normally would. It might slow them down a little bit, but they have some degree of resistance. Okay. The solution to this, at least temporarily, is chemists sort of fought back by saying, well, what if we make an inhibitor that blocks the beta-lactamase enzyme? And so you have a beta-lactamase inhibitor that will have to be administered along with the antibiotic. And so now you've got the antibiotic that's actually killing the bacteria, and then you have the second inhibitor that's used to turn off the resistance enzyme. And so it's sort of this constant fight back and forth. The problem is some bacteria have evolved a second variant of the enzyme that can destroy the beta-lactamase inhibitor. I vote for it being called beta-lactamase inhibitorase, which you can see where this is going. Bacteria then will very likely develop another enzyme that will destroy that inhibitor, and then we'll have to make a new inhibitor, and it'll just keep going back and forth and back and forth. It's going to lead to the point where you'll have to administer so many inhibitors, it's either going to stop being effective, or it's just going to be too toxic because the drug dose will be too high. And so it's a really good illustration of how natural selection takes place. The other important thing to point out is the gene for this, and I mentioned it before, but just to really reinforce it, that beta-lactamase gene, that didn't just pop up recently, right? We only know that bacterial antibiotic resistance has been around since the 50s in a clinical setting, but it's been around in nature, in soil microbes, and in other organisms probably for a very, very long time, many millions of years. And so this beta-lactamase gene isn't really new, it's just new to human pathogens, probably passed by a plasmid or a virus or some mechanism like that. Right? So that means administering the antibiotic didn't cause this enzyme to come into resistance, but it certainly has acted as a selection pressure to encourage it to spread and become more common. Right. The next handful of drugs that we're looking at, they have their effect by blocking protein synthesis, and blocking protein synthesis means interfering with ribosomes or mRNA in some way. And I'm not going to go into a lot of detail on these. What I want you to see is it's possible to have a lot of different drugs that target the same structure, because a ribosome is actually a pretty big, complicated molecular machine. Okay? The reason they make a good target, prokaryotic or bacterial ribosomes are fairly different from eukaryotic ribosomes, and so the binding sites for the enzymes or the inhibitors don't line up. That means you get a pretty good therapeutic index. Toxic to the bacteria, very effective, but not so toxic to the person, and so you have more flexibility. And the first in the example that I'm going to use, aminoglycoside drugs are a pretty a commonly used category of broad-spectrum antibiotics, all bacteria have ribosomes, and so they'll target just about any bacterium that you expose to them. And the way they have their effect is they bind to and change the shape, distort the structure of the ribosome. The 30S thing, if you're curious, ribosomes have two subunits. The 50S is the big one, the 30S is the smaller one. And if you distort the shape of the ribosome, it can't read mRNA correctly. And so if you remember that table a while back, that code that showed you these three RNA nucleotides code for this amino acid, it causes misreadings. It essentially makes the RNA blurry, if you will. And so the ribosome can't read it correctly, which causes translation errors. That just means mutations. And so you get proteins that have the wrong amino acid spliced in, which causes so much dysfunction inside the cell, the bacterial cell. It's still making protein, but they're all incorrect and not doing their function correctly. And so very toxic, cell dies. Tetracyclines still mess with ribosomes, but they take a slightly different route. Instead of causing mutations, they just block the transfer RNA. Remember, that's the molecule that actually carries the new amino acid into the ribosome. And it acts like a competitive inhibitor to block the tRNA binding site. And so whereas aminoglycosides caused a bunch of errors in protein synthesis, tetracyclines just flat out block it. You can't make protein in the first place. Cell metabolism shuts down, and cell dies. Macrolides are another group, still targeting protein synthesis, but a slightly different strategy here. The idea is the ribosome is still correctly shaped. It can still read the RNA. The tRNA can still bind, but that last step in the translation process, linking the two amino acids together, amino acids are connected by a peptide bond, it blocks them from actually forming, connecting amino acids to each other. And you end up getting the same result as with tetracyclines, right? You can't make new protein. And so what I want you to see from these three examples is you've got a lot of options, right? These big molecular machines, these enzymes, like a ribosome is a gigantic enzyme, you can block it in a lot of different ways. And I only picked three examples. There are quite a few more, actually. The one group of drugs that I mentioned earlier, nucleic acid synthesis blockers that don't work very well. I mentioned this before. The same concept, it's the same concept as chemotherapy. It's very easy to use inhibitors to block DNA replication or RNA synthesis and transcription, but they're toxic to humans. And it's because polymerases are too well conserved. Conservation in an evolutionary sense means they haven't changed much. And so bacterial DNA polymerase is just so similar to human polymerase, it's hard to find an inhibitor that blocks one and not the other. The effect is they're toxic to bacteria, but you've got a pretty poor therapeutic index because they're also toxic to humans. All right, the last thing to consider here is, do antibiotics have side effects? And I put this in here just to show you measuring side effects in a drug, any drug, antibiotics included, is incredibly difficult. And one of the reasons that it's difficult is if you have big, obvious, and widespread side effects, then they're easy to detect. So for example, a lot of antibiotics mess with gut resident microbes and can lead to GI dysfunction, discomfort, diarrhea, things like that. Somewhere around 10% of people, especially children that take antibiotics, will have those GI symptoms. That's easy to detect because it's so common. Others, this is an example of a category of antibiotics that causes a fairly small but still detectable risk for retinal detachment. And if you look at the numbers, what you'll see is the risk isn't enormous in an absolute sense. 3.3% in antibiotic treated versus 0.6% in controls. And so the numbers are pretty small, but that's still 5 times as common in the antibiotic group. And so it's definitely having an effect, but the problem is if no one's specifically looking for it, right? No one probably thought, oh, I'm going to look for cases of retinal detachment in people that are taking these antibiotics. It's very hard to detect. And so when you have a lot of side effects that are happening at a very, very low percentage of patients, it's really hard to tie them together. And so side effects are always underreported by definition because it's impossible to find them all. This is one reason why antibiotics shouldn't just be used indiscriminately. You have to say, is it worth it? Is the infection bad enough? Is the risk to the patient bad enough that we're willing to give them antibiotics knowing there are probably some side effects that we're unaware of, and also that the antibiotic is likely to encourage resistance. And that's really the last topic in the notes. I'm not going to go through all this resistant stuff again because I did in the last video. You can go check it out from the previous section if you need a refresher. But I want you to be remembering this resistant stuff. Remember what antibiotics can and can't do when they're acting like a selection pressure. And to sort of add on to what I've said in the past, this is just data showing you, and we'll talk about it in class, antibiotics are very, very widely overused. They're prescribed for viral infections where they won't have any effect. They're prescribed for bacterial infections where the benefit is so small that it's probably not worth it. And just sort of think about why is this happening and what can be done about it. And all I'm trying to get everyone to think about is benefits versus cost. If the cost is a lot of uncomfortable side effects and the encouragement of resistance, and the benefit is maybe makes you a little more comfortable than you would have been, but isn't saving anyone's life, is it really worth doing that? There's been a movement lately that antibiotics should be treated as not a complete last resort, but maybe given a little bit more respect for how powerful they are, and not just throwing them at any old infection that comes along.